Hi everyone, we're back here at the NJIT campus / Rutgers, which is right across the street there. But if you want to follow along with everything, feel free to tweet us at Scicomm Monday. I'll try to talk over the sirens that are going by right now. And then the hashtag Rutgers NJIT Scicomm is the hashtag we're using for all of the broadcasts and everything that's going on here during this Scicomm mega trip over to the East Coast. And then if you want to follow up later with me, if you've got any other questions, feel free to tweet me at my personal handle at wildlifebiogal.com or wildlifebiogal.com. That's my website. It's a long day of broadcast, so which I'm excited that we're doing something completely different because this will keep me on my toes with everything, which is always good. So we're going to switch gears from all the ecology side of stuff, and we're actually going to be talking about some cell and molecular biology. So this should be fun. So for all of you micro folks out there that like all the cool things that are more like that indoor science with your microscopes and all that, this is going to be your fun time. So I've got Huri Mücahit. I was so close, man. I got the J in it though. I was good. Yeah. But so why don't you tell us a little bit about your academic background so people kind of know like your experiences coming into what got you to where you are today.
Okay, sure. So just so we can go over again, it's actually Huri Mücahit.
Oh, did I get that wrong?
That's okay. It's all good.
We're going to restart the broadcast and we'll be...
Not a problem. Trust me, this is 20 some odd years of this, so. So yeah, I am a third-year PhD student studying cell and molecular biology, and I kind of, took a route, I guess, of a typical cell biologist to an extent, in that I started off not thinking that I was necessarily going to go into cell biology because, the only thing I knew about biology was really just the health sciences. So I really thought I was going to go into medicine. But after working as an EMT and all that, I realized that really wasn't the route for me. After graduating college, I took a couple of gap years, tried to figure out essentially what it is that I wanted to do. And afterwards, joined the master's program here, took the cell molecular and developmental course offered by one of my advisors actually, as well as another professor, and I kind of found love. So as cheesy as that sounds. And then thankfully after working with a couple of different professors, I was able to join the PhD program. So here I am.
Right. Yeah. So why don't you kind of bring us up to speed with your research, because I know being an ecologist on that side of things, they don't deal a lot with that cell and molecular biology side of things. So why don't you bring us up to speed, and myself as well. So I'll just sit here like the side, like the rest of the crowd with you.
No problem. Sure. So basically, so I work in two different laboratories, and both of these labs essentially work on similar and different things. So one of the labs I work in is looking at how a cell skeleton rearranges when cells migrate, especially when they migrate collectively.
And so what is a cell skeleton for those of us who don't know what that is?
No problem. So a cell skeleton, cytoskeleton, there are three different types of cytoskeleton, but the main purpose of these is to provide structure, rigidity if necessary for cells, and to allow for movement. And they serve a couple of different other functions as well. But
so we have a question coming in. So the cells that you're working with, are they human, plants, or animals?
So we work with animal cells. We use that as a model for essentially what's going on in our bodies as well.
Okay. Are there any specific species of animal cells? Are there certain lines that you guys tend to work with, or is it just a wide variety? Or So the first lab I was talking about, they work with a frog, Xenopus flavus, specifically frog cells, during the first 24 hours of development. And the second lab that I work in, we work with canine kidney cells. So they're a pretty well-established cell line called MDCK cells. Okay. So tell us, keep going with the whole cytoskeleton and everything, and moving on from that of
Sure.
bridging our education to where you are right now.
So yeah, as I said, there are 3 different cytoskeletal systems. There's actin filaments, microtubules, and intermediate filaments. So this first lab that I work with, Dr. Barber's lab, that lab focuses on intermediate filaments, specifically keratin intermediate filaments, which are basically the proteins that provide the most structure. They're essentially like the steel rods of your cells. They're also basically what allows for your nails and your hair and all that. And the second lab that I work in works with another cytoskeletal system called actin. And that's where we work with the cell lines. And we look at, again, specifically the how cells adhere to each other and how the cytoskeleton allows for that.
Right. So like if you have any questions, feel free to send those and can get your education in cell and molecular biology. It's your 30-minute PhD right here. This is so we have a question coming in. How do you predict about human cell interaction on the basis of frog cells? Which is a good question. Like, how do you make those leaps from different animals to what the human cells are doing?
So the first 24 hours of development in many ways are very similar to human development. In addition, a lot of the proteins that are being expressed are very, very similar to, and in some cases, really the same proteins that we see within our human cells. So we use these, again, as a model.
Are there like any big differences? Like, like is it mostly so similar during that 24 hours that you don't really notice any differences? And then like once you get out of that 24 hours, like what are like the big differences that you start seeing that makes it maybe not so good of a model?
Sure. So they are similar in many ways. Like I said, the proteins specifically that we study, these keratin 8 and 18 proteins, they are basically what we study in human cells, and really they're the same. They've been known to be the same. The frog cells, after the first 24 hours, you definitely start seeing some differences, and especially if you let them go for a couple of days, you then start seeing the cells, becoming, these organisms becoming tadpoles eventually and all that, so.
So we had a question coming in. Why frog? Like, why is that like a model species for you guys to use?
Sure. It's because it's a little bit simpler. So there are a couple different species, and a lot of the different labs work with different species. So like I'm just gonna say dragonflies based on the other labs. Fruit flies, right?
There you go.
Exactly. Sorry.
Yes, I don't have to feel so bad about mispronouncing your name now if you can't remember one. Yes.
Yeah.
I feel better now.
It's not a problem. After talking with a whole bunch of people from Ware Lab, right? But so there are fruit flies, mice, rats, a couple different organisms. So the reason frogs were chosen within this lab is because they are a much simpler version of essentially what's going on within our own cells, right? So for these keratin proteins, there's only 2 of these keratins that are being produced within the cells, versus say a human cell may have many, many versions of that, many isoforms.
So it's just like doing that simple model that breaks it down and you can see the basis of it, versus like having something that's so complex that you're not going to be able to keep track of where everything is moving in the cell per se.
Yep.
So is another reason, like a question came in, are the frogs easy availability? So is it like a species that there's just a numerous amount of them, it's easy for the labs to obtain, is what I'm assuming that they're trying to go for with that question?
Yeah, to an extent. I mean, yeah, we can definitely get those.
So, no problem. So tell us like what all of these actually we had another question come in that's better than my question, so we'll ask that. So how do you separate proteins from the frog?
So you use a lot of biochemistry, honestly. A couple of different processes. I mean, you run these cells essentially through different buffers and what have you, and basically it separates out DNA, it separates out RNA, proteins, and what have you. And then from there we can run them through Western blots is one assay that we can use, and it basically separates out your different proteins based on weight or size.
Okay.
So
a follow-up question. It looked like what organ specific. So I think they're so asking which part of the frog are you getting that from, or has it like not differentiated out into organs yet when you're working with it?
So yeah, definitely no organs yet. There are general regions that we work with. So specifically, we work with the mesendoderm, and eventually, how do I say
You're the expert, not me, so like,
okay.
I'm not even going to try to jump in on that.
It's all good. So basically, this would be like, eventually these can go on to form organs, but right now this is very, very initial stages.
All right.
Yeah.
So it's kind of like there's like these masses of cells, but they aren't like technically organs yet.
It's just a ball of cells
at
this point.
So I guess well, we got another question coming in, and
Wow,
like I said, these are better than my questions, so I'm totally good. And I'd much rather have the engagement of the people online. So and of course, I totally just missed the question.
How do you function? Of
yes,
the
there you go.
I
Yes,
think it was.
So
right. So as I was saying, keratin proteins essentially are part of the cytoskeleton or the cell skeleton, right? And like I said, these are like the steel rods of your cells. So they really make sure that when your cells are tethered together, right, they can really remain tethered, because you don't want your cells ripping apart, right? Especially when they're
will they literally
moving.
just kind of like just disintegrate if you didn't have like those steel rods there, or just like when you say like break apart, like I guess like it's providing that structure of which like the cell can then adhere to and then stay like in that shape, or
yeah, so it definitely provides structure, and it definitely allows for cells to adhere to each other as well as to their surfaces. So cells can definitely break apart, and it's not obviously the worst thing because it happens, right? But it's more so when cells, for example, need to move together. So during the first stages of development, you have cells that are migrating and shifting and all that. So you obviously want to make sure that the cells that are designed so eventually become, say, your eye, right, don't necessarily go down to by your feet, for lack of a better example. Right, so.
Yeah, so we have a question here. Is it affecting on cell level or molecule levels or organ levels? So I'm not 100% sure where they're going with that question. So
if I can ask for a clarification,
send in a clarification of that question. We'll definitely appreciate it. We'll get back to that. So when you're looking at all this stuff, so kind of tell us maybe a little bit like the equipment that you use to do all of this science. So
sure,
let us like mentally imagine that lab, kind of like walk us through that.
No problem. So in order to be able to do this, we have a couple of different rooms essentially in each of these rooms to house different types of equipment. So one room will be devoted specifically to the microscope that we use, and what's really cool is we can fluorescently label certain proteins and look at them underneath the microscope and basically watch these cells move live. And I have a couple really, really nice videos of that too. Others are, we also sometimes will fix cells and look at a variety of proteins and how they may potentially be interacting together. Right, so a lot of microscope work, obviously, because it's much more difficult to see without it. Now, in addition, we also have all of our cells obviously need to be kept in a sterile environment to prevent any type of contamination. So we have a culture room.
Like, what kind of contamination would you actually see in those cells?
Bacteria.
Okay.
Bacteria, and also contamination by other cell lines.
Okay.
So if you're working with something that's very specific, you don't necessarily want another cell line getting into that.
Okay. Oh, so we have a clarification coming in. I mean, on which level that protein acts, so what group of cells makes molecules and which then groups together from organ?
Sorry, I'm
going to let you see if you can figure it out more than me.
So. You mean on which level do proteins act? And I mean, so the proteins essentially, proteins work on cellular level, right? So you can have cell adhesion proteins that are what ultimately anchor the two cells together, right? Okay. You can have proteins essentially that are involved in trafficking and sending signals throughout the cell, right? And then eventually within one cell, because this is happening, say, within cell 1 and cell 2 and cell 3, this can eventually have an effect on an organ level as well.
Okay. Yeah, hopefully that answers the question there with things. And I see Canada has joined in, so hello Canada. It's good to see you. So anyways, so how do you deal with like that contamination, like what's the best way to keep like the bacteria and those other cell lines from getting into what you're actually looking at?
A lot of alcohol, a lot of ethanol everywhere. Making sure you have just what they call the BSL-1 type safety gear, you know, so like your gloves, lab coats, you know, depending on what you're working with, sometimes you'll need goggles and all that, so.
Does it ever get like where you have to go beyond that basic safety gear with what you're working with, or are you still just at that, like, you know, what you said, what was it, BS1?
BSL-1.
BSL-1, okay.
Uh-huh. I TA micro as well. So,
okay.
So this is some of the stuff that we work with, yeah. No, thankfully, none of the stuff we work with we necessarily have to worry about, because these are just essentially the same cells that make up our organs.
Okay.
Right? But when I like TA micro, sometimes you obviously would have to worry about that.
Right, it's not like you're dealing with, you know, some of the diseases and other fun stuff that I'm reading about in the book Spillover right now, so it's not Ebola that you're having to worry about.
That's terrible.
That would be like every safety gear possible, please. Thank you. So I guess what's some of like the really neat things that you've been finding with your research of how these proteins interact and help with the cytoskeleton?
So one of the things I think that really surprised me and a lot of people, and is still considered a relatively new aspect of the field, is how interactive each of these different cytoskeletal systems are. You know, so technically we say, okay, so there are 3 cytoskeletal systems, right? There's actin, there's intermediate filaments, there's microtubules. But each of these is physically linked to each other, and they adjust accordingly. So for example, if you pull on one, you'll see an effect sometimes on the other, right? Mm-hmm. So I think that was, when I first saw that definitely kind of like blew my mind.
I guess which is your favorite of the 3 systems? Like if you had to like pick a favorite, like which one's the most interesting to you?
So speaking honestly, when one lab works on one and the other lab works on the other, I don't know if I would necessarily feel comfortable choosing a favorite. You know, I think both systems are really great.
Okay.
So.
It safe, dang it. So what's the, like we always ask the ecologists, like what's the biggest fieldwork fail that people have had? So I guess what's the biggest like lab fail that you've had that you can share with everyone?
So this is not my story necessarily.
Yes, let's rat out other people.
And I would definitely not name this person, but one person almost accidentally set a system on fire.
Yeah, that would be a slight fail.
And it unfortunately, fortunately he was there, but if he had not been there, it would have been when nobody else was in the building. So it could have been pretty
burning
terrible.
down like your school's biosciences buildings. Oh, it's really good. Yeah. So we had a question come in asking if this was the field that you always wanted to get into, or like do you have other interest in science?
So I'm gonna flat out say no. Absolutely not.
Hey, I mean, I have to admit, like when I went into grad school, I wanted to study cougars. That was like my ideal thing, and I talked to a couple different professors. I said, if you study cougars in Michigan as a grad student, you're going to ruin your career right away, because there was argument, you know, are they in Michigan or not? And so I ended up going into Great Lakes and working with swans, and I'm really happy that I did. Like, I mean, it's led to some really great experiences, but if like you asked me like when I first started, oh, you get excited to go work with swans, I'd be like, say what? So I totally feel you on that.
Yeah, I mean, I was definitely interested in a lot of basically trafficking within cells as well, and I worked with another professor who really helped me out, but after rotating with a couple of different labs, I realized that cytoskeleton, part of the reason cytoskeleton is the most interesting to me, part of the reason is because you can, it's nice to see this really, really striking image of basically like a whole bunch of fibers, a whole bunch of what look like holes in some instances going through your cell, right? Oh. So that was really cool for me.
I guess if there's anything else like you could study, like what would be like your like goal study thing that you would do, like, you know, once you're done here, like what would be like what you would want to study after this? like, would you want to stay working with cytoskeleton and how they function, or would you like want to like branch off to like, hey, this sounds really interesting, I'm gonna go do that?
Well, I'm kind of pretty terrible with this and I say this because every time I go to any conferences, I'm like, oh wow, that's really interesting. Oh wow, that's so cool. So I think it's really interesting to look at. So basically we have DNA, RNA, and protein, right? I mean, this is essentially what's established. So I think RNA signaling and the factors that allow for RNA to be translated one place versus another, I think that's very interesting. And then as an aside, one thing I was considering, which is totally out of left field, but I was really interested in ecology too.
You want to go study like fish in the ocean or something?
But then I realized I would have to be out in the field, with dealing with all of everything else that comes with ecology. So it's like, maybe not.
Yeah, I have to admit there's some fun aspects to being out in the field, and there's also some things that it really is like, can anything else go wrong with everything?
Yeah.
So what's the difference between DNA and RNA is a question that's coming in.
So DNA is essentially what we typically think of when we think of, the genetic material within your cells, right? What essentially allows your cells to become, what allows your cells to become what they are and eventually form organs and eventually form you, right? So DNA is a much more stable version of that. So RNA is typically what is thought of as the material in transit, right? So you would use the stable genetic material to transcribe or basically form RNA off of that, and then from there you form proteins.
Okay. Right, and then we had another question come in asking, can you edit cells with precision? And I don't know exactly what they're talking about, so I'm totally leaving that up to you.
So I'm gonna assume you mean the genetic material within cells, and yeah, there's actually a lot of really, really cool work that's going on out there. Some of the work that we do, honestly, is including a fluorescent tag for various proteins that we want to work with, so that's definitely something that you can edit, right? Others is to remove or to knock down, essentially, or a gene, knock out a gene.
Yep, so we had a question come in, is DNA unique to everyone? So, like, does everyone have, like, a unique set of DNA and, I guess, RNA as well as that? So,
so I think, honestly, you would be able to answer that, too, as far as your ecology background goes, just in terms of differences between various organisms and all that.
But
right?
you're the guest that is going to be the expert on this, so like I'm just asking questions. I'm totally letting you have it.
Sure. So I mean, yes, there's definitely a unique component to every person's genome, right? I mean, there's 99% similarity?
It's something along that. I know I heard the number way back early before.
Yeah. I mean, in many ways, we're pretty much the same thing, but with unique aspects, that essentially make you.
Right. And we had another question come in about cloning. So, like, is it possible to be cloning adults? And I'm assuming they're talking about humans, but maybe, some of the animals you work with. And I have to admit, I'm a big fan of Orphan Black, and it's all about clones. And so, like, if you're, like, geeking out about science, that's the sci-fi show that you should definitely watch. But, yeah, have you had, like, any experience with cloning organisms? Like, and do you know, like, any of the background with, like, are you allowed to do that? Because I know there have been cloned organisms, but, like, how common is that, like, studied, in cellular molecular biology?
Okay.
I just threw a whole bunch of questions at you.
No,
I'm sorry.
So I'm, like, trying to be like, okay, so where do I start? Okay. So, when you typically think of cloning, you immediately think of just making a carbon copy of yourself, right? And obviously we're not at that stage right now. But we do obviously do a lot of bacterial cloning. We use that essentially to produce specific proteins that we want.
How hard is it to do that? Like, what's, like, the process kind of to do that?
Hard, okay. Yeah, you basically add in this plasmid of DNA that you want into the culture, and through just a couple of steps, you know, like you agitate the bacteria and all that, right? And eventually it incorporates it into its own genome, and thus you have multiple things.
Like, how long is the time process for
Not
that?
very long,
like,
honestly.
I mean, like, does it take like a half hour to do that, or 5 minutes, or like 10 hours, or?
I mean, typically you want to give the bacteria time to essentially grow and multiply as well. So, I mean, let's say the entire process is going to take like a day.
Oh, okay.
You know,
That's not too bad.
yeah, it really isn't. Yeah, and I mean that's allowing for time to grow, you know,
Okay.
and all that. So,
Yeah. So, I mean, there is cloning possible, like, but it's not quite up to the level of being able to clone a human adult yet, or at least
definitely
that
not.
we
Yeah, because there's
know
so many
of.
things
Yeah.
to consider. You know, there's so many variations, and you know, control, I guess, of gene expression is very complicated. You know, it's definitely not just so you. In simple models, we can see effects of, so say we knock out a specific gene, we can see an effect for it, you know, but some genes you can't knock out because then the organism doesn't survive, right? You know, so there's a ton of things to consider. I did see one question actually before about grad school life.
Yeah.
I think it was.
Yeah.
And grad school life it's hectic, it's crazy, but it is a lot of fun at the same time. You know, it's just, as I'm sure any grad student can tell us,
I mean,
anyone.
I would not be sitting here today if it wasn't for the fun, hectic nature of grad school, but the benefits that you get out of it are really cool because you get to meet all these amazing people that,
seriously.
you know, growing up, you're like, these scientists are so cool, and now you actually get to hang out and talk to them and learn from them and get to do some exciting work with them, which is always fun. Yeah, so... I know we had another question that came in is, you know, do you work with human cells? And so like, it seemed like earlier you were talking like you work with frogs and dogs, yeah, the kidney cells of the dogs.
Yeah.
Is there, so like, one, do you work with human cells at all? And then like, if you were, like, are there issues like because I mean, I think, what is it for? Is it humans? Is it the IRB that you have to get permission from to be able to work with any sort of human beings? I know it's like it's IACUC for us when we work with animals.
Yeah, there's definitely a lot of regulation, obviously, when it comes to human cells. Obtaining permission to initially get those cells. You know, you can definitely work with human cells. My work specifically has not involved any of that, but if necessary, I can get it. And a lot of times it's from patients, you know, so patients who have specific cancers, you know, say breast cancer, you know, kidney cancer, you know, any one of those. So.
Yeah, because I was interested to see like how you can take those models of what you work with and then how do they really actually translate to what happens in human beings, because it's so complicated to be able to do that. And obviously, you know, like you're speaking of cancer, I mean that's one of like those big buzzwords of people, like they are like, oh, cancer, and it's like it's a very scary thing, and so like any research that we can get out there to be able to help prevent that or like cure that is always, you know, quite of interest, you know, with many people out there. So we've got a couple minutes left here, so make sure that you're getting in any of your last minute questions here. So if you want to learn more about cells and cytoskeleton, all the cool stuff that happens in there, and proteins and things, or grad school life or life here in the metropolitan area of favorite scientist. Yeah, I like that question. So yes, what is your favorite scientist?
Ooh, okay.
It's always a tough one because I have so many. It's like, okay, which one do I say is my favorite?
Exactly.
Like,
I mean, it's kind of a tough one. I mean, there are people who really made an impact in the field. So one person, and I as soon as I mentioned this name, and my lab mates here, they're all going to laugh at me because they know about it. You know, but I love like Kathleen Green. Her work is just so pivotal. She has so many publications on what I'm studying essentially. Let's see. You know, I don't even know.
Yeah, like I think I have like
the
different
spot.
levels of favorite scientists. Like you know, say like Jane Goodall is like one of like the most amazing people, and hearing her come talk at Central Michigan University a couple of years back was so amazing. Like I was totally geeked out. Versus where like I have like more like academic level folks that I really love. Like Dr. Pat Chow Fraser from McMaster University was one of my like early like fandom, like, oh, she's just the most amazing scientist, and be able to get to know her and hang out with her lab now has been one of the most exciting experiences, you know, I've been able to have. And like, you know, I have so many of those, like, you know, different levels. Like I have Scicomm, like, fandoms, like getting to meet Dr. Michelle LaRue from University of Minnesota. I got to meet her last year at Wildlife Society, and she was just as amazing as you hoped she was, because she was just so positive and being, you know, wanting to help like everyone out there in science. So there's so many great levels of different scientists, and I have fans of, like, you know, other grad students that are doing great work and seeing them. So it's like, it's such a hard question to answer, because like I'm just naming a few, but it's like I have this huge list. It's, you know, not quite one of those teen bop, you know, boards where like, oh, these are all my favorite scientists, but it's still kind of like, in a way, you know, is that, because there's just like, there's people that there's the work that you really respect, and then you also respect them as people as well, not just the science too. Like, I think that's always what really impresses me, is when they're not just a good scientist, but they're a good person as well, and that's like what really endears me to that. So
yeah, I have to say, honestly, as cheesy as it sounds, you know, and again, my lab mates are gonna make fun of me, I think my own advisors are really, really brilliant scientists, and I'm so lucky to be working with them. And it's, if you look at any of the universities, any of these scientists who work there, it blows my mind how brilliant so many of these people are.
Yeah, definitely. It's like, you know, I know I'm not the smartest person in the world. I know I'm not the dumbest person in the world, but it still like amazes me, like the level of like thought process that people have, and they always like make me up my game when I hang out with them, which is so good. And like, I'm always learning more things by hanging out with all these folks, which is so much fun. So one last question we'll answer here, and then we're gonna have to sign off, is, do you want to have your own lab someday? So like, would that be like an end goal for you? Like, do you want to stay in academia, or do you want to like go outside of that and have your own lab, like more on like the private or maybe like a public sector, like outside of academia, or do you want to stay in academics for you?
So I actually, one of my favorite things, honestly, of being in this university is working with students, you know, and getting to show them essentially what we do. So I love doing that. I also really love my research, and I love the fact that we get to learn new things. So I think I would definitely like to continue academia.
Yeah, there is something cool, like being able to teach students and then like kind of watch that light go on in their eyes when they get it. It's just like, it just makes you feel so good, because you're like, you know that they're so excited because they totally got it or learned something new, and it's just like, it just makes you proud, like to be able to like help them out with that. So with that, we're going to sign off. If you have any other questions that we didn't get to, because I know there was a whole bunch that started flying in, and I'm sorry we didn't get to them all, feel free to tweet them to Scicomm Monday or to my personal handle at Wildlife BioGal, and I'll make sure they get through. Follow all the tweets that are happening with the Rutgers NJIT Scicomm hashtag, and we have I think it's the last broadcast coming up with Stephanie at 3:45. I keep wanting to say 4. No. I'm wrong. I'm totally wrong with the times. I've totally messed up. I was so off. 4:15. 4:15. Yes, because I was a half hour behind with my times. So yes, with that, we're gonna sign off. Thank you definitely for all the fun with cellular. Like, I learned a lot, which is really great, and I hope you all learned a lot. I hope you had fun.
It was a lot of fun. It was nice to see all the questions too, because some of them I was like, okay, let's see how do I explain this.
Yeah, if you guys are seeing this on a replay, definitely feel free to send your questions in via Twitter, and we'll see you live here in a few more minutes with Stephanie to wrap up the Rutgers NJIT. I've been saying it all day. I knew I'd finally mess it up. Scicomm fun here on Periscope. So until then, you guys have a good one.